All right, let's dive in. Today, we're decoding the very blueprint of life itself. We're talking about nucleic acids. You know them as DNA and RNA. These are the incredible information molecules running the entire show inside every single living cell on this planet. It's going to be a fun one.
But first, let me start with a puzzle for you, a real head-scratcher. If you could take all the DNA from just one of your cells and stretch it out, end to end, it would be over 7 feet long. So, how on earth does all of that information get crammed into a space so small you can't even see it? Just keep that question tucked in the back of your mind. We are definitely coming back to it.
So, here's our roadmap for this explainer. We're going to start with how these molecules were even discovered. Then we'll break down the structure of DNA piece by piece. After that, we'll explore its different shapes, meet its super versatile cousin RNA, and then, we'll finally solve that packaging puzzle. We'll wrap things up by looking at how scientists can actually test the code's integrity.
Okay, so where did our understanding of all this even begin? The journey to unlocking the secrets of DNA is, it's one of the great scientific detective stories of the 20th century. It's filled with brilliant minds, a few rivalries, and some truly game-changing discoveries. This story actually kicks off way back in 1869. A guy named Friedrich Miescher first finds this mysterious substance he called nuclein, but what it did? Total mystery. It wasn't until the 1940s that experiments by Avery, McLeod, and McCarty finally proved that DNA was the stuff of genes. The final, absolutely critical clue came in the 50s from Rosalind Franklin's incredible X-ray images. They were the key that let Watson and Crick finally solve the puzzle, revealing that iconic double helix shape in 1953 and, boom, launching the entire modern era of molecular biology.
All right, so that's the history lesson. But what is this famous molecule, really? Let's zoom way in on the DNA blueprint and see what it's made of, one building block at a time.
First things first, let's clear up a little distinction that trips a lot of people up: the difference between a nucleotide and a nucleoside. A nucleotide is the whole package. It's got a phosphate, a sugar, and a base. Think of it as the complete Lego brick. A nucleoside is just the sugar and the base. So the key takeaway is simple. Add a phosphate to a nucleoside, and you've officially got a nucleotide. Easy, right?
So every single one of those complete building blocks, the nucleotides, has these 3 parts. You've got the phosphate group. This is the part that links them all together to form the strong backbone of the chain. Then you have a 5-carbon sugar, which is actually where the name nucleic acid comes from. And finally, the star of the show, the nitrogenous base. This is the part that carries the unique genetic information, the A, T, C, and G.
Now, here's something that is so cool. Check out the tiny difference between the sugars in DNA and RNA. Just look at that 2' carbon. DNA sugar, deoxyribose, is missing an oxygen atom. See, it just has a hydrogen there. RNA sugar, ribose, has a full hydroxyl group, an OH. Now that might seem like a tiny change, but it makes RNA way less stable and much more reactive than DNA. And that's perfect for DNA's job as the rock-solid, long-term information archive.
Next up, those bases. They come in 2 families, purines and pyrimidines. The purines, adenine and guanine, are the bigger ones. They have a 2-ring structure. The pyrimidines, cytosine, thymine, and uracil, are simpler, with just a single ring. And remember that key difference. Thymine is strictly for DNA, while RNA swaps it out for a very similar base called uracil. So, how do all these individual blocks link up to form a chain? Well, the cell calls in a master builder, an enzyme called DNA polymerase. It creates this super strong connection called a phosphodiester bond. It links the 5' end of one nucleotide's phosphate to the 3' end of the next one's sugar. This makes a chain with a really clear direction, kind of like a one-way street with a 5' start and a 3' finish. And when you bring in the second strand to make the helix, they run in opposite directions. We call that anti-parallel.
Now that we have these 2 anti-parallel strands, what's holding them together, zipping them up down the middle? That's the job of hydrogen bonds. And this is where it gets interesting. The connection between guanine and cytosine is extra strong. Why? Because it's held together by 3 of these hydrogen bonds.
On the other hand, the pair of adenine and thymine is held together by just 2 hydrogen bonds. This difference in bond strength might sound small, but it has huge consequences. It means that regions of DNA rich in G-C pairs are much tougher for the cell's machinery to pull apart, which is a big deal when it comes to replicating and reading the code. So, we've got our double helix, but believe it or not, that's not the whole story. DNA isn't just one single static shape.
Depending on what's going on in the cell and what the sequence is, it can actually twist and turn into a few different forms.
The classic shape you always see in textbooks. That's bDNA. It's the long, thin, right-handed helix, and it's the most common form. But you've also got aDNA, which is a bit shorter and broader, and you see it when RNA is paired up with DNA. And then there's the real weirdo of the group, zDNA. It's a left-handed helix with a cool zigzag shape that tends to pop up in areas with lots of G's and C's.
Now what happens when you introduce some tension into that helix? Say when the cell is trying to replicate it and has to unwind it. The DNA gets all stressed out, and one way it relieves that stress is through something called supercoiling. The analogy here is perfect. It's exactly like twisting a rubber band tighter and tighter until it's forced to coil back on itself.
And you know what's wild? We can actually describe this twisting with math. The equation is L equals T plus W. Don't worry, it's simpler than it looks. L is the linking number. Basically, how many times the strands are linked, and for a closed loop of DNA, this number cannot change. T is the twist, which is just the normal turns of the helix, and W is the writhe, which is the number of supercoils. This little equation shows us that if you change the twist, the DNA has to compensate by changing the writhe by supercoiling to keep that linking number exactly the same.
All right, let's move on and talk about DNA's incredibly talented cousin, RNA. You see, if DNA is the master blueprint that's kept safe in the office, RNA is the multi-talented contractor that's out on the job site getting all the work done.
Let's put them right next to each other. As we've seen, DNA is typically a double-stranded helix, uses deoxyribose sugar and thymine, and it's built for stability, perfect for long-term storage. RNA, though, is usually single-stranded, uses ribose sugar and uracil, and is far more reactive. This allows it to fold into complex shapes and take on a huge variety of active roles in the cell. And man, look at all those roles. You've got mRNA, the messenger that carries instructions from the DNA. You've got tRNA and rNRNA, which are essential for actually building the proteins. But amazingly, some RNAs, called ribozymes, can act as enzymes driving chemical reactions. And then you have others, like sRNAs and microRNAs, that are key players in controlling which genes get turned on and off. RNA really is the jack of all trades of the molecular world.
Okay, time to go back to our big question from the beginning. How do you cram 7 feet of DNA into a microscopic nucleus? The answer is an absolutely brilliant system of spooling and packing. It's a masterpiece of biological engineering. It happens in multiple levels. First, the negatively charged DNA wraps itself tightly around these positively charged proteins called histones. This forms a little unit called a nucleosome. This first level looks kind of like beads on a string. Then those nucleosomes coil up into a thicker fiber, which then compacts even more. This process of coiling and looping just keeps going until you end up with the incredibly dense, tightly packed chromosome that we can actually see under a microscope when a cell is dividing. It's just a phenomenal feat of data compression.
So for our final section, let's get practical. If DNA is this precious, irreplaceable blueprint, we need a reliable way to know if some chemical or substance out there is causing harmful changes or mutations to that blueprint.
One of the classic ways we do this is with the Ames test. It's a really clever setup. You start with a special strain of bacteria that has a mutation. It can't make its own supply of an amino acid called histidine, which means it can't grow without it. You then expose some of these bacteria to the chemical you're curious about. Now, if that chemical is a mutagen, it's going to cause new, random mutations. And just by pure chance, some of those new mutations will actually reverse the original defect, fixing it. These newly fixed bacteria can now grow on a plate that doesn't have any histidine. So the bottom line is this: the more colonies that pop up on your test plate, the more mutations the chemical caused. It's a pretty clear sign that you're dealing with a mutant.
So there you have it. From the simple bases that form its code to the elegant way it twists and spools itself into a microscopic space, the double helix is truly life's master blueprint. We've learned to read it, to understand its structure, and even to test its integrity. And all of that leaves us with a final, really exciting question. Now that we can do all this, what is the next great frontier just waiting to be discovered inside our own genetic code? Thanks for joining me.